Hi, everybody. I'm Vincent and I want to address a document that was posted to Zenodo some time ago called Unusual Features of the SARS-CoV-2 Genome, Suggesting Sophisticated Laboratory Modification Rather than Natural Evolution and Delineation of its Probable Synthetic Root. This is a 26-page document. It has not been peer-reviewed. It has not been subject to official scrutiny by virologists and other scientists. The authors are from the Rule of Law Society and the Rule of Law Foundation in New York. And this document has been widely criticized by scientists everywhere. It suggests a laboratory origin of SARS-CoV-2, which of course isn't correct. And it looks very imposing. The authors have PhDs. One has an MD and a PhD. There are lots of charts and data. However, the scenario for the laboratory origin of SARS-CoV-2 is completely implausible, as stated in this document. We recently discussed this on TWiV, This Week in Virology, and I just want to play the excerpt of that episode in which we discuss some details about this document. I would suggest the manuscript, The Proximal Origins of SARS-CoV-2. All right, now finally, the last document that we have here. Many listeners.
Document's a good word.
A document, that's what it is. A document was sent to me by a number of people yesterday, and many listeners got at least 20 different emails saying, you need to address this. And I really find it unfortunate that we have to, because it's just going to take time that we could do more productively with something else, but we have to do it.
Plus, it gives it air, which we prefer not to do, but it's getting air already.
Yeah,
so
and people want to know, because as many people say, this looks okay to me. What do you think? I'm not a scientist. You know, it's a 26-page document with lots of words and figures and data, supposedly, and it kind of looks authoritative. But as you...
Oh, it looks very authoritative. We got diagrams, we got models of the spike protein, we got nucleic acid sequence, we got mutations, got the whole shebang.
We got a reference section, although when you go to look at it, about 50% of the things or more are just preprints or a website. Or
lots of websites.
Yeah. So it is a 26-page document which is called Unusual Features of the SARS-CoV-2 Genome Suggesting Sophisticated Laboratory Modification Rather than Natural Evolution and Delineation of its Probable Synthetic Root. And the authors are Yan, Kang, Guan, and Hu, and they are from the Rule of Law Society and Rule of Law Foundation, New York, USA. And there's an email there that you could email the authors from. And, you know, just reading the title, I mean, this is not a title of an article that would ever appear in a journal. And as the abstract and the introduction is just written in a way that just says everything else is nonsense and what we're going to tell you is really what happened. And I want to go through it in a little bit of detail, but as we'll tell you, the data are not at all conclusive. It's really nothing different from what we talked about months ago, and in which has been debunked in published articles and so forth. And that's why it's unfortunate that we have to go through this. But a kind of an interesting thing where you can just put out what's apparently a definitive-looking document, and some of these authors are apparently scientists. I think the first author was a former coronavirus person. I don't know what this society or law foundation is.
Actually, I have some information on that, but we can come back to that.
All right. So people have said to me, please give it fair consideration. So we'll give it a fair consideration. All right. So basically, the idea here is that this virus was engineered in a laboratory, and they purport to give evidence for that. And I will just say up front, there's no evidence for it whatsoever. There's more evidence that this came from nature. We've talked about this multiple times on TWiV. There's really good evidence. What they have here are accusations that are not founded in any way. And we will go through and we'll talk about it. And let me just a few ideas from the abstract in the introduction. They say, despite its tremendous impact, the origin of SARS-CoV-2 has remained mysterious and controversial. No, it's not mysterious and controversial. We have wonderful evidence, genomic evidence, that it came from a bat isolate and that is a recombinant of many different viruses, and the lineage tracks back decades. We did a paper not too long ago showing that. It's very clear. So this is a way of using words to kind of start to, get in between your sensibility and say, oh, maybe they're right. The natural origin theory, although widely accepted, lacks substantial support. I don't know. Every virologist I know is okay with it. I don't know who you're talking about here.
That statement is not true.
Right.
The alternative theory that the virus may have come from a lab is censored on peer-reviewed scientific journal. No, it is not censored. It is rejected because all the manuscripts don't have any support for it. Isn't that a horrible way to say
peer review is?
Censored? I mean, and to a person who's not familiar with the scientific process, they always say, "Oh my God, they're being censored? Really?" No, it's just peer review.
In my typical way, I'm going to be pedantic about the writing. I wanted to point out earlier that the title would be better with a comma after the word evolution because it's two independent clauses. And here, the first sentence about the natural origin theory, and the alternative theory is censored. They're not even parallel. Yeah.
There are lots of writing issues here.
It's a problem. But they're trying to get to people who don't know about the science and use these words, and then people are like, yeah, I guess that could be right. They say the virus has biological characteristics that are inconsistent with a naturally occurring zoonotic virus, and they go through it, and we'll talk about that. But that's not true. It has very consistent properties. And they point out two isolates, so two bat coronaviruses that were isolated in 2018, ZC45, ZXC21. They think these were the template or the backbone for making this virus. And I'll tell you why that's totally improbable, unlikely. And then, of course, they say we have to investigate. In the end, they say the lab should be investigated for sure because we have to prevent this in the future. So there's some, in my view, because there's very little scientific support for their idea, I think there's something else behind this paper that I don't understand. The introduction is also full of this. And then here's one thing that really is annoying. This is a Nature Medicine paper that we talked about a long time ago by a variety of authors, including Rambeau and Eddie Holmes and Ian Lipkin and others. The proximal origin of SARS-CoV-2, where they go through all the genomic data and they say this, for all these reasons, had to come from nature. It couldn't have come. And they say here in the introduction, the central conclusion of this article is now being challenged from scientists all over the world. Baloney. No, it's not being challenged.
Not true.
And then they say the authors of the article have conflicts of interest, raising concerns on the credibility of it. All this is a smokescreen, in my opinion, right?
Not only that, but that's not science.
No. Right. And I think it was Newsweek that pointed out that the conflicts of interest are that Ian Lipkin received some award from some Chinese institute. And then the second one is just pointing to Eddie Holmes' CV.
Yes.
So
this is true.
Yeah. Then they say all of the evidence that this is from nature, the virus is based on this virus called RATG13, right, which we have talked about for a long time. It's an isolate from 2013 from Yunnan, a cave in Yunnan, which has 96% nucleotide sequence identity with SARS-CoV-2. And they say this virus is bogus. Actually, later on, they say it's fake data put out there by scientists to mislead them. Okay. For which I see no evidence whatsoever. But I tell you, 96% identity is higher than the identity of the two viruses that they can claim are the backbone of SARS-CoV-2.
And I think that this is actually a really important piece because they say that both RATG13 and the one other virus that seems to cluster closely in the Nature Medicine paper are both fake.
Yep.
And so they completely discount them. And that is why they're going with these alternatives, ZC45 and what is it, ZC21. But the other reason why I think that this is actually a really important point is that later on in the text, they mention some more of these issues about RATG13, and they say that they are in the process of putting together another manuscript on RATG13 and how it is fake and such, and that manuscript is forthcoming. And I point that out because that makes me think that they are going to get another news cycle when their second manuscript comes out. And so we should sort of be aware that that is coming and that does not have any support. And so we can sort of preemptively say that there is not evidence for that.
So the one of the ways I look at this is that the difference between science, one of the differences between science and pseudoscience is that in pseudoscience, you start with the conclusion that you want, and then you cherry pick the data to support that conclusion with a complete unwillingness to change anything about the conclusion. In real science, you may start with a hypothesis, and then you examine the data in an unbiased fashion. And if the data don't support your hypothesis, you modify the hypothesis, and then it's a reiterative process. You go through that. Now, in this case, being pseudoscience, okay, they have their conclusion that this thing is fake. And in order for that to work, they have to say that the RATG13 virus is fake, because otherwise their hypothesis doesn't work. As we've already said, there's no evidence for that, but they have to throw out that virus, okay? Otherwise, the whole thing crashes.
You know, what's amusing is that RATG13 was originally supposedly the backbone of SARS-CoV-2 until people pointed out, 4% difference is a lot of bases in a 30,000 base genome. It's not likely. And they're scattered all over. Why would someone change them all to SARS-CoV? It doesn't make any sense. And now they're picking two viruses, which they say are the backbone. These ZC45 and ZXC21. And by the way, RATG13, its provenance is very clear in the literature. You can find the original isolation. Peter Daszak talked about it. His organization was involved. There's just zero evidence that it's fake or manipulated in any way. So I discount that completely. But their viruses, ZC45 and ZXC21, I found the references to them. They were both identified separately in 2018. And it's true that they are from institutions with a military affiliation, okay? And they say because they were made or isolated in these military universities that they're nefarious, right? So that alone is saying that they have to be the back, but they must have manipulated these. But these are, in fact, bona fide isolates of viruses. But the interesting thing is they have even less nucleotide identity with SARS-CoV-2. One of them
Which is why you have to throw out RATG13.
Yeah, so one of these two is 89% in a genome of 30,000 bases. That's 3,300 nucleotide differences. There's no way that's the backbone. There's no way someone started with that and somehow changed the receptor binding and a few other things and got to SARS-CoV-2. Sorry.
Yeah, I mean, later on, they talk about a proposed way that one could have done this. And that sort of implies, well, that's where all these base pair differences come in. But then they have other places where they're very strong about how base pair differences didn't happen. And so it's sort of like there are 4 that totally happened, or the 4 that totally didn't change, but these 3,300, they changed.
Yeah, I should point out that we talked earlier about SARS-CoV-2 is changing about 20 bases a year, right? So how many years would it take to have 3,300 change? A lot of years, right? 10, 100 years or something. So there's just no way that this is a valid argument. And it's, I think that you can't convince any scientists of this, but maybe people who don't understand the material would say, okay, it sounds good. So that's one thing. The second thing is they focus on the receptor binding motif, which has a very high affinity binding to ACE2. It's very different from SARS-CoV. It's a better binder. And of course, the bad viruses don't have high affinity binding site for ACE2. And they say this was made. This has to have been made in a lab because it's such high affinity binding. And I would say, well, that's why it wasn't made in a lab, because in models that were made previous to SARS-CoV-2 of the binding of the RBD to ACE2 based on SARS and other coronaviruses, these amino acid changes would never have been predicted to increase binding. It would be something else, and no one would ever have done this. And as had been pointed out, if you tried to do it in cell culture, it would take you years and years and years to do this. So for the very reason that they're arguing that it had to be made, that's actually why it wasn't made in a laboratory, because no one would have known how to do this.
Yeah, so they sort of have this argument that this receptor binding motif of SARS-CoV-2 doesn't bind as well to bat. And so it could definitely not have come from bat because it binds so poorly to bats. And there would never be a virus in bats that has a receptor binding motif that binds poorly to bats. But I think that one thing people should also think about is that this is a part of the virus that tends to change a fair amount, because this is the part where antibodies are binding. And so there is actually some selection for variation here. And in fact, it's quite likely that you would get changes in the virus, some that might bind better, some that might bind worse to bats in the bat as the virus is trying to avoid the bat's immune system.
I also think that these high affinity changes just arise randomly. And we talked about this with Raul Rabadon. They arise randomly because it doesn't matter for the virus in bats if you have high affinity ACE2 binding. And then if this virus happens to encounter a human, then it replicates very well, and maybe an additional change is selected, who knows. But there doesn't have to be a selection for this high affinity binding site.
You raise another question that I was going to pose, and that is, we don't really know when this spillover happened.
No,
right?
We don't. No.
Okay, so it's possible. Correct me if I'm wrong, but it seems to me possible that in fact it spilled over decades ago, and some of the maturation of affinity for the human receptor happened in the human population.
It could be, sure. It could be, for sure. We don't know, but that's why we need to do more sampling, and apparently this is not being done very much these days, so that's a problem. And then the final thing, which is a key part of their argument, they say there's this furin cleavage site in the spike. It's a cleavage site for a cell protease, right? They say this is definitely put in by a human because this is not in any bat coronavirus, and this is key to reproduction in human cells. Well, that statement is wrong. There's a paper that we discussed a while ago in Current Biology describing this isolate RMYNO2, which also has high nucleotide identity with SARS-CoV-2. And I quote from the last sentence of the abstract. Critically, and in a manner similar to SARS-CoV-2, RMYNO2 was characterized by the insertion of multiple amino acids at the junction site of the S1 and S2 subunits of spike. This provides strong evidence that such insertion events can occur naturally in animal beta coronaviruses. So their statement that furin sites are never found is wrong. And that's a key part of their argument. And then finally, one last thing. They say, oh, look, there's a restriction site on either side of the gene for the receptor binding domain. They could have used that. Yeah, the restriction sites are everywhere. It doesn't mean a darn
And in
thing.
this day and age, if you really wanted to be doing engineering, you probably wouldn't be doing it with restriction sites.
That's right. You'd be doing
more
PCR,
modern
right?
ways.
Yeah,
I mean,
for sure.
Three of us might choose to do it that way, but Brianne would jump
us
on
old
some
guys,
other newer technique.
I would use a Gibson assembly, which would not leave any restriction sites. And I think that they're sort of a couple things to say. One, if having restriction sites in a genome mean that something is engineered, then E. coli is engineered.
Oh no! Are you accusing that, Brianne?
As are most microbes and most organisms, but the restriction site thing, I think, is a really important part of the cherry picking, because earlier they say, oh well, those 3,300 mutations in the backbone just happened during passage.
No,
but somehow the restriction sites absolutely didn't mutate. And there are also 2 codons that they say are unusual codons somewhere else. And those managed to not mutate, but 3,300 other random things totally did.
Yeah, this is not... And, you know, the thing it's unfortunate is that apparently this first author has some virological experience. And so for her to say these things, it's either intention or ignorance. I don't know which one. It's very sad because
hard to know what the motivation is, except that you get airtime.
I don't know what the motivation is here. But anyway, so basically all these arguments were made before. They're not correct. There's nothing new here. And I think you should take it as another attempt at getting attention. And I'm not sure what the motive is, but it's not correct. This virus came from nature. It didn't come from any laboratory.
Well, I have a little bit of, I consulted a friend of mine who might have some, I thought might have some insight into both the organization, the affiliation that these people talk about, and the individual herself. And if I could just spend a moment on that. He says he doesn't know anything about the foundation. It appears to be one of the organizations founded by Miles Gao, Wengui Gao, a shady Chinese businessman, and he gives a Wikipedia link to it that's actually quite interesting. I read that. Gao's photos prominently displayed on the website. Gao is a crook and had become a billionaire real estate developer in China through shady business practices, including using bribes and taking advantages of a close connection to the Chinese intelligence agencies. After Xi came to power and the Chinese officials who were close to Gao lost out in the power reshuffle, the Chinese authorities started to investigate the business dealings by Miles Gao. He narrowly escaped China and then turned into an activist, first against some Chinese officials and then the entire Chinese government. He has a YouTube channel, and his videos are full of rumors and lies about his enemies, which are now mainly the Chinese government. Before Steve Bannon was indicted, Bannon stayed in a yacht owned by Gao and appeared frequently in Gao's YouTube videos. I don't believe anything from Gao or any organizations associated with him. And in fact, he's later, this friend of mine later sent a link to one of these videos with the author talking on one of these on one of these YouTube channels, talking in a sort of what appears to be a news format, and this is the ultimate in fake news, talking about how hydroxychloroquine is actually a miracle drug. And Bannon's on the same YouTube video.
So tell us who Bannon is, Rich Condit.
Steve Bannon was a political advisor to the Trump campaign, and ultimately hired by Trump after he was elected as a strategic advisor for the first 8 months until he fell out of favor. And Bannon has done all sorts of things. Actually, he has a military career early on. Ultimately, he's had some business in the financial sector. He's ultimately got a Harvard MBA, but ultimately landed him, created a sort of alt-right quote news unquote service called Breitbart News. Okay, which is basically and as I said, an alt-right, shall we say, information or disinformation outlet. So
would that be truthiness?
Who knows what motivates these guys? But Gao, it seems to me, from what everything I can tell, is basically a crook who got caught by the Chinese and now sees on a vendetta against the Chinese.
I see.
Okay. With respect to the author, she was educated in mainland China, both an MD and PhD, and had been a postdoc in a very reputable coronavirus lab in Hong Kong until a few months ago. She started to, quote, reveal, unquote, supposed true nature of SARS-CoV online and caught the attention of a U.S.-based blogger with a close connection to Gao. At the urging of the blogger, she moved to the U.S. To expose the supposed cover-ups by the Chinese government and her Hong Kong employer. She came to the U.S. without her husband of 6 years, who works the same Hong Kong lab with her, but does not hold the same views of her. She's since appeared on Fox News and some YouTube channels backed by Gao. My friend says you didn't pay too much attention to what she says because she really didn't have any firsthand knowledge about COVID-19. So this is just crazy stuff to the max.
So listener Tim had sent in similar stuff, and he said this paper was originally posted on Zenodo, a website which is basically a publicly available repository of science and academic research to which anyone can upload their work. Both of the nonprofits behind the study were formed with Guo, with whom Bannon has collaborated on a number of advocacy efforts targeting the Chinese government and business endeavors that have drawn the scrutiny of federal law enforcement officials.
So the Rule of Law Foundation is a 501c3, which is a type of nonprofit. And then there's the Rule of Law Society, 501c4. And Steve Bannon serves as the chairman of the 501c4.
Beginning to suss what's going on here.
God.
And the website of the organization says their vision is to permit the people of China to live under a national system based on the rule of law, independent of the political system of the People's Republic of China. And that its mission is to expose corruption, obstruction, illegality, brutality, false imprisonment, excessive sentencing, harassment, and inhumanity pervasive in the political, legal, business, and financial systems of China. So nothing to do with science, infectious disease, etc. Just, yeah.
This is strictly radical politics.
Yeah.
Should we say keep your politics out of our science?
Yeah.
Yeah. Yeah.
So this is nonsense, folks, and the authors. I think that you're doing a disservice to the world. The only thing I can imagine is that you were paid for this in some way, but I'm sorry because this is not how science works, and you should be ashamed of yourselves. In fact.
And if people want to hear anything else about the science, if they look at the details on the in vivo fitness parts of this, they are just wild.
Oh, yeah. Actually, that was another comment that I was going to make about this because, you know, they go through all of this complicated stuff about how you would engineer this virus. And then they talk about various passaging experiments in animals and other things to make it fit for transmission and have appropriate pathogenesis characteristics in humans. And it's total nonsense, complete nonsense. And this to me is actually, it's an important thing about the whole, the pandemic in general. We don't know what makes this virus transmissible in humans, really. As transmissible it is, we don't know what controls the pathogenesis. And so if you wanted to create something that had the characteristics of this in humans, you wouldn't know how to do it. And passaging it in animals is not going to do it, okay? That's going to give you fitness in that animal, but probably not necessarily, and probably not, fitness in humans. So that's all nonsense.
It's just replete.
Nonsense is an understatement for this section.
You know, we've already put more time into this than we should, but I do not think anyone should think about this for another moment.